Welcome students to the genetics series as we look into another module on this series. While here, don't forget to like, please pass a comment, share with others and subscribe to the channel so that you can always be updated when a new topic is out. Today we'll be going straight into module 3, and this has to do with exceptions or departures from the classical Mendelian ratios. So on normal situations, the expected Mendelian ratio of 3:1 and 9:3:3:1 is expected. But there are situations where these ratios do not hold. And when this happens, we refer to it as departure or exceptions from the classical Mendelian ratios. Now, the major causes of such departures are incomplete dominance. Now, in incomplete dominance, two alleles may produce an intermediate phenotype when both of them are present, rather than one of the alleles fully determining the phenotype of the offspring. Now, this mostly occurs when a dominant allele does not completely prevent the expression of a recessive one. As a result, it results in partially marked phenotypes, which are usually intermediate to the two extreme phenotypes, that is the homozygote, the dominant, and then also the recessive. We can draw examples from the coat color locus of cattle and also from the flower colors of the plant Mirabilis jalapa. In most progenies arising also from marriages involving different races. Okay, so in cattle, the allele capital R for redness exhibits incomplete dominance over the small r allele for whiteness, such that in the F2 generation, the RR, that's the dominant homozygote, the dominant heterozygote individual, and the recessive homozygote individuals are respectively red, brown, and white in color. And this will give us a phenotypic ratio of 1:2:1, not the 3:1 that is expected. And this is a clear departure from the expected Mendelian ratio. To show it diagrammatically, these are the parental gametes. So in the F1 generation, all will be red. This is signifying that redness is actually dominant over whiteness. Now, when we serve the F1 individual, these are the gametes produced, and this is what we get in the F2 generation. We have one that is red, two of them will be roan in color, and one will be white. If you look at the example of the color alleles of Mirabilis jalapa plant, such that the red also does not completely, the red flower color does not completely dominate the white. It will give us a ratio of 1 red, 2 pink flower, and 1 white in the ratio of 1:2:1. So there's 1 red, we have 2 pink flower colors, and then there's 1 white. And all this as a result of incomplete dominance by the dominant red color over the whiteness. Another reason for departures from Mendelian ratio is what is known as lethality or the influence of letter genes. Now, these genes are found both in plants and animals, and where they are found, they could lead to deformities or even death. Now, if it is death, it may occur at any stage of the development of the individuals that are carrying the letter genes. Likewise, also the deformities. So, these are examples of letter genes in animals, farm animals. Creeper, wingless, missing upper beak, and shaker. These are all found in birds. In calves, you have hairlessness. The calf is born without hair. In pig, you have atresia ani. You also have imperforate anus in pig and sheep. You have cleft palate and amputated. These are found in many of the domestic animals. Therefore, when lethality is in operation, it can lead to deficiencies of certain genotypes or phenotypes in the population. We'll take an example from a mating of two creeper fowls. When two fowls carrying the creeper genes are mated, it only results in two genotypes. Why? The reason is that the dominant homozygote embryo carrying this genotype usually dies off within 3 to 4 days in the egg post-incubation. So as a result, this phenotype does not appear in the F2 generation. So only the heterozygote individuals and the recessive homozygotes are found to hatch at the end of the incubation period, such that the phenotypic ratio is 2:1 and not the expected Mendelian ratio of 3:1. So this creeper is a case of a dominant letter, that is, the effect of the letter gene is felt when it is in the dominant state. There are also situations where the effects are felt when they are in the recessive state. So we want to show an illustration. These are two creeper fowls. These are the F1 gametes, and when you mate them, the dominant homozygote dies during incubation, within 3 to 4 days of incubation. And then what you end up having is a phenotypic ratio of 2:1. The heterozygotes survive, and likewise also the recessive homozygote. Epistasis. This is also one of the reasons for departures from classical Mendelian ratio. Basically, epistasis is an interaction between non-allelic genes. Non-allelic genes are genes that are usually represented by different alphabets, and they could be found on the same or on different chromosomes. A non-allelic epistasis will lead to the masking of one non-allelic gene by another. One of the genes will prevent the expression of the other. So we want to take an example. In poultry, the color producing gene is represented by capital C, and this color producing gene is masked or prevented from expressing itself by the color inhibiting gene, capital I. So we want to consider a mating between a dominant white leghorn with this genotype and a recessive colored plymouth rock with this genotype. When you cross these two individuals, what will be produced in the F1 will carry the genotype this: capital I, small I, capital C, and small c. So when we intermate the F1, we'll get the following results and progeny. So these are the gametes of the F1, and intermating will produce these progenies. So anywhere there is capital I, it will prevent or mask the expression of its non-allele, which is capital C. such that the resultant F2 generation will be in this ratio. You have about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 that will be white. There will be no appearance of plumage, while you have 3 of them that will be colored because of the influence of the capital C gene, color producing gene. So this is the explanation of what happened here. The presence of the dominant I, that's the color inhibiting gene, will always prevent the expression of capital C, which is the color expressing gene. Now, this doesn't matter whether it is in the dominant state also. The obtained ratio we get is 13 white as against 3 colored, which clearly is a departure from the expected ratio of 9:3:3:1. Now, why? We want to know the reason why this occurs, or it's like this. The reason is anywhere the capital I is dominant, it will prevent the appearance of color in the plumage because it is an inhibiting gene. And even where there's recessive is in the recessive state, small I, small c, small c. The small I will also prevent the small c from expressing itself. And that is the reason why even these birds are having white coloration as against being colored. So the other forms of epistatic interactions, and we'll quickly look at them. We have the dominant epistasis. This occurs when the dominant allele A of one allelic pair will express itself even in the presence of another dominant or recessive allele of the other allelic pair, which is capital B or small c. So in this case, it is only when the genotype of the individual is homozygous at the epistatic locus that the alleles at the hypostatic locus can be expressed. Thus, because of this, the genotype A underscore B underscore D underscore and this will produce the same phenotype, while this and this will also produce 2 additional phenotypes, such that the observed F2 ratio is 12:3:1 and not the 9:3:3:1 expected. Now, the hypostatic locus represent the alleles that are being dominated. Okay? So let's look at an example here. We want to assume that when an A, that's the dominant dark, A is dominant dark, represent dominant dark, small a is recessive white, B is dominant long tail, and small b is recessive long tail. So the gametes we obtain from the F1 when a dominant dark and long tail is mated with a recessive white and short tail animal, rat in this case, will produce this phenotypic ratio. So wherever you have this, it will block this from expressing itself. So we end up having 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 that are dark and are having long tail. And then we have this 1, 2, 3 that are white and long tailed. And then we have this which is white and short tailed. So the ratio is 12:3:1. We have a case of recessive epistasis, and this will occur if the recessive genotype at one locus, for example small a, suppresses the expression of alleles of another gene, either in the dominant or at the B locus, either in the dominant or in the recessive state. Then the allelic pair, when that happens, we say that this allelic pair, small a, is showing recessive epistasis over B, meaning that the small a is preventing the expression of even the capital, the dominant non-allele that is in combination with. So alleles at the hypostatic D locus can only be expressed when capital A or this combination is present. So because of the genotype, this and this will produce two additional phenotypes. And this ratio 9:3:1 becomes the ratio of 9:3:4. So we are going to take an example again from the previous examples. I mean, in terms of the dominant and recessive alleles. So when a cross like that happens, because of the recessive epistasis, we have 1, 2, 3, 4, 5, 6, 7, 8, 9 will produce. Then we have this, and then we have this represented. So we also have a case of duplicate genes with cumulative effect, and this happens when the dominant conditions, either in the homozygous or the heterozygous state, at either of the locus, but not at both of the locus at the same time, will produce the same phenotype. So the F2 ratio in this case becomes 9:6:1 instead of 9:3:3:1. The genotypes, this and this, will produce the same phenotype. This genotype will also produce another different phenotype, while this will produce yet another. So let's look at an example. So you can see the phenotypic ratios that came up. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, as against 1, 2, 3, 4, 5, and 6. 5, and then 1. 9:5. 9:6. So duplicate dominant epistasis. This occurs when the dominant allele of both loci produces the same phenotype. In this case, the dominant allele this is epistatic to capital B and small b. Also, the dominant allele capital B is epistatic to capital A and small a. So the 9:3:1 ratio is modified into a ratio of 15:1 in this situation. So this is an example of the cross. Okay, you can see because of that epistatic reaction, we have a ratio of 15:1. Sorry, please, you cannot see this. Let me see if I can move the video. Okay, this is not possible. But you have a ratio of 15:1. Then we have duplicate recessive epistasis. In this case, the allele, the recessive allele AA is epistatic to capital B and small b, and small b is also epistatic to this and this. So identical phenotypes are produced by both homozygous recessive genotypes. Wherever this is present and this, they'll produce identical phenotypes. So because of that, this genotype, this, and this will produce one phenotype. When both the dominant alleles are present, they complement each other, producing yet a different phenotype. So the obtained ratio is 9:7 instead of the classical 9:3:3:1. This is an example. We have 1, 2, 3, 4, 5, 6, 7, 8, 9. We have 1, 2, 3, 4, 5, 6, 7, 9:7 ratio. So polygenic inheritance is also one of the reasons for departures from the classical Mendelian ratio. Polygenic traits usually are controlled by several genes, with each of them acting at several loci, and each of them makes contribution to the overall phenotype of the progeny. So let's consider, for example, a mating or a crossing in wheat. This is the gene for wheat color, and this shows that wheat color is a trait that is controlled by 3 different genes acting as several loci. So we have A, B, and C being the 3 different genes. So how many individuals will be obtained from this cross? We expect a total of about 64 different individuals, all of the genes interacting differently. Now, the possible F1 genotypes we get will be A as given here, A, B, C, capital A, capital B, small c, and so on and so forth. This is A, B, C, like that. So, because there are so many gametes, it will not be very easy placing individuals into distinct classes because of the interactions of these different genes and also because of environment and genotype interaction. So the classical Mendelian ratio is not possible under such situation. Another reason for departures from the classical Mendelian ratio is linkages. Now, genes that are found in the same chromosome are said to be linked, and they are usually separated by physical distance, which is measured in map units, or what is referred to as the Morgan. And linked genes are usually transmitted together. And how easy will it be for them to be transmitted largely depends on the distance between them. So linkage affects Mendelian ratio basically because when linked genes do not separate independently of each other during gamete formation, and because of this, it may lead to disequilibrium in the number of gametes formed. It is this disequilibrium in the gamete that leads to phenotypic ratios that are completely different from the Mendelian ratios. So this illustration is to show us crossing over. And this crossing over occurs at the early stage of meiotic prophase. Here we are looking at it combines the two chromosomes of the pair into one. The process is called genetic recombination. So the exchange of genetic material between two homologous chromosomes will lead to the production of a new genetic combination called recombinance. And like we said before, this depends on the distance between the genes on the chromosome. The farther the distance, the easier it is for crossing over to occur. So let's consider the gene loci W, capital W, small w. For white, as the capital W represent white skin, and the small recessive W represent yellow skin in chicken, and the NA, capital N, small a, this for naked neck, this is naked neck, and this is normal feathered neck, chicken, the chromosome of the chicken. So when there is no crossing over, there are only two possible gametes that will be formed from this chromosomal interaction. And at this stage, we refer to them as non-recombinant, non-crossover because there's no crossover. And it is safe to simply call them the parental gametes. So we want to assume a situation of a single crossover between the two gene loci, as shown here. So this is prior to crossing over. You can see them arranged in their normal way. But with crossing over, you see that crossing over occurred at this point. This is the product after crossing over. So the small na has shifted up while the capital N and small a are shifted down. So what we have here is a recombinant. This is a case where the crossing over is at three different gene loci, 1, 2, 3. So it will take this form. Maternal inheritance. There's another type of inheritance pattern known as maternal inheritance, and it leads to departures from the classical Mendelian ratio. Embryos are usually formed due to the fusion of male and female gametes, and in most species, the female gamete is larger than the male gamete, and it provides the cytoplasm for the developing embryo. Within the embryo, there are certain factors which are released by nuclear genes of the female, primarily the mitochondria in animals and the chloroplasts in plants, which specifically affect the developing embryo. So the cytoplasm plus the mitochondria, they have DNA which controls certain traits in the offspring. In most plants and animals, the mitochondria and the chloroplasts are strictly inherited only from the female, and that is what is referred to as maternal transmission, due to the fact that the male gamete, that's the part that takes part in reproduction, is devoid of them. So traits that are controlled by cytoplasmic DNA of females are said to express maternal effect, and the inheritance pattern is known as maternal inheritance. So we want to take an example from the snail, where the nature of the shell coiling in the offspring is determined by the DNA in the cytoplasm. Also in the 4 o'clock plant, Mirabilis jalapa, remember this plant? The branch color may be normal green, pale green, or variegated due to 2 types of chloroplasts. So the inheritance follows maternal inheritance approach because the type of pollen grain, the male, which is the male sex cell of the plant, has no effect on what the branch color will finally be. So because of this, we say that it follows maternal inheritance. So where the male gametes do not contribute much to the phenotype of the progenies, there's every likelihood that there will be departures from the classical Mendelian ratio. The number of gametes required may not be complete, as a result there will be departures. Another reason for departures from classical Mendelian ratio is sex linkage. Genes carried on the sex chromosome, like the X chromosome of humans, show a different inheritance pattern than genes on the non-sex chromosome. And as a result, sex linkage goes against Mendelian ratio. So why? We want to know the reason why. This is because while the females have the XX chromosome, that is, they have 2 alleles for a trait, the males have only 1 X chromosome, therefore only 1 allele for the trait. So in crosses involving X linkage, the genes involved are carried only on the X chromosome. The Y chromosome does not carry a superscript, and this is because it does not have genes that are homologous to those that are found on the non-homologous portion of the X chromosome. In this region, only a few genes actually have been identified. Among them are the testis determining factor, which promotes the development of the male phenotype. So the Y chromosome is also shorter in size and contains fewer genes when compared with the X chromosome. Examples of X-linked traits in humans are red-green color blindness and hemophilia. The eye color in Drosophila, the fly, is also another example of an X-linked trait. So the pattern of inheritance of X-linked trait is special because males do not have a second copy of the X chromosome. Therefore, when there's any defect on the X chromosome in males, there will be no copy of it that would have masked the effect for showing. Therefore, the disease linked to it mostly occurs in male offsprings, and this disturbs classical Mendelian ratios. Codominance. In codominance, 2 alleles may be simultaneously expressed when both are present. The major difference between codominance and incomplete dominance is that while in incomplete dominance, the dominant allele is not able to completely dominate the recessive allele. In codominance, each of the alleles is capable of some degree of phenotypic expression in the phenotype. So an example in the shorthorn cattle, when a brown coat cattle with this genotype is crossed with a white coated one with this genotype, the F1 progeny, which is heterozygote, is found to possess roan color as against brown or white. And this roan color is such that both the brown and the white hairs occur in definite patches on the animal's body, but no intermediate color is found, as is expected in incomplete dominance. So this is a clear departure from the Mendelian ratio, because what is expected in the F1 generation is a situation where the dominant allele dominates the expression of the recessive allele, and as a result, all the F1 progenies will carry the phenotype of the dominant gene. But this situation does not occur like that. There's interspacing, and each of the colors of the parents are represented in the F1 heterozygote. So the implication is that when you carry out a cross in the F2, you are not likely to get a ratio of 3:1 or 9:3:3:1, and this is a clear departure from the Mendelian ratio. Another reason found to also affect Mendelian ratios from holding true is what is known as multiple alleles. Recall that Mendel studied only 2 alleles in the guardian P, but in many real populations, there are often more than 2 alleles of a given gene at work at the same time. And a typical example is the ABO human blood group. Now, in the ABO blood group, there are 4 different types of phenotypes or blood groups. These are the A, B, AB, and O. Meanwhile, these are produced by 3 different alleles, represented as I capital A, I capital B, and I O. Now, the allele stands for antigen A, while this stands for antigen B. Both this and this are codominant. That is, they cannot dominate each other. And in people having the blood group AB, however, both of them show dominance over those carrying IO blood group. Therefore, 6 possible combinations are possible from the interaction of 3 alleles. We can either have this, or this, this, or this, this, or this, respectively. Therefore, individuals with this or this, we have blood group A. Individuals with this or this, we have blood group P. These individuals are having blood group AB, while these have blood group O. Now, because of this kind of relationship, it means that Mendelian ratios will be very difficult to obey. So, we have come to the end of this module, and now we know the reasons why departures from classical Mendelian ratios can happen in a population. Thank you for listening. I'll wait to see you in our next module. Meanwhile, don't forget to like, pass a comment, share, and subscribe to this channel. See you in the next